Welcome to My Hometown, the program that explores clubs, organizations, businesses, and issues across Nassau and Suffolk counties and sheds light on the different towns that are making a difference.
A great day to you and welcome to another edition of My Hometown on the voice of Nassau Community College 90.3 WHPC. I'm your host, Bill Haran, along with Nassau Community College student Zach Turkel. Our special guest today is Dr. Adrian Krainer, a professor at Cold Spring Harbor Laboratory right here on Long Island. His lab studies the mechanisms of RNA splicing, ways in which they can go awry in disease, and the means by which faulty splicing can be corrected.
In particular, they study splicing in spinal muscular atrophy, which is a neuromuscular disease that is the leading genetic cause of death in infants. The Krainer lab is currently using similar approaches for the study of other diseases caused by splicing defects. The Krainer lab has also worked to shed light on the role of splicing proteins in cancer.
Recently, Dr. Krainer was featured in Newsday as he will split one of the most lucrative awards in science, a $3 million breakthrough prize for his research and the life-saving medication to treat spinal muscular atrophy. Welcome to my hometown, Dr. Krainer, and congratulations on your award.
Hi, thank you very much.
Now, Dr. Krainer, first of all, let's begin. Tell us some of the details of this amazing award and a little bit about yourself and your background.
Okay, where should I start with the award? It's the Breakthrough Prize. It's been in place for a few years, and it's sponsored by Silicon Valley philanthropists. And the awards are in life sciences, but also in physics and mathematics. And they pick several laureates every year, and it's a somewhat secretive process. And so then the awards are given around November, and there's a big event that surrounds this with a lot of celebrities, and it was a lot of fun.
Do you get a lot of high fives and slapping hands and crowds cheering?
Yeah, it's quite a production. So they refer to it as the Oscars of Science, and it pretty much looks like that with a red carpet and a lot of photographs are being taken. A lot of Hollywood celebrities participate, and altogether it's a lot of fun. And there's videos that are put together to explain each the scientific aspects of every award, and then people step to the podium there to receive the award and say something for half a minute or so. And the following day there's a symposium. And it's usually done at Berkeley, Stanford, or UCSF, and this year it was at Berkeley.
So we're dealing with a real science rock star here today, right?
According to them, yeah.
We're honored to have you on the show. What brought you to Long Island, the Cold Spring Harbor Laboratory?
Yeah, so when I finished my PhD, I was a student in biochemistry at Harvard, and I knew of Cold Spring Harbor Laboratory because there's annual some meetings in a bunch of areas. For my field in particular, there was an annual meeting on RNA, and so I had already come here as a student, I think in 1984. And so they recruited me to come after my PhD in 1986, and that's what I did. So this was really my first and pretty much only full-time job for 32 years or so.
Dr. Krainer, Bill mentioned earlier in your bio about RNA splicing. Is RNA the same as DNA?
It's not the same, but we consider these molecules chemical cousins. So they're both nucleic acids. So that's a type of biopolymer. And so DNA is the genetic material, and RNA serves in part to transmit the information from our genes to the products, which are typically proteins. So also one difference is that DNA has two strands. It's the famous double helix, whereas RNA is single-stranded.
Now, Dr. Krainer, we use the term splicing RNA. What does that mean? I mean, for most of us, when we cut something, or we're just talking about physical things like a loaf of bread or meat or something. But when you're doing it in the scientific world, and most of us aren't privy to that, what does that mean? What's involved?
Yeah, people do get confused because there's a term gene splicing, which applies to recombinant DNA. That's kind of a technique where we put together bits of DNA from one species and to introduce it into another. But RNA splicing is a natural process, and it's a kind of editing or cutting and pasting. Essentially, if you have a strand of RNA, it has parts of it, segments that have important information, and those are separated by other segments that are non-coding. And so splicing is a cutting and pasting whereby all the unimportant bits are removed and then the important coding segments are joined together, pretty much as one would edit film. So you have a long film with important scenes and others that the director decides to cut out. So you have to make two cuts to remove the part that you don't want and then splice the remaining segments of the film together. So that's the analogy.
I'm guessing
also if you splice cable, that would also be somewhat of an analogy.
But is this done? I'm guessing this is microscopically done. Is that correct?
Yes,
tiny,
of course.
Tiny.
So within each cell in our body, in particular in the nucleus of the cell, there are many RNA molecules coming. They're copies of different genes, and each of them has to undergo this cutting and pasting in order for the genetic information to be properly interpreted.
The prize you recently won, Dr. Krainer, had to do with the disease spinal muscular atrophy. So what is the disease and its symptoms? And until now, there was no treatment or cure, right?
Right, yeah. So spinal muscular atrophy, or the acronym SMA for short, is a neurodegenerative disease. It affects motor neurons in the body, a little bit like Lou Gehrig's disease, which is more famous. Does, but there's a different genetic basis, and SMA primarily affects children, sometimes very young infants, and it leads to the gradual death of the motor neurons in the spinal cord. And motor neurons are nerve cells that go from the spinal cord to the different voluntary muscles in the body, and so the result of this degeneration is that there is a weakness. Primarily of the legs and arms, and this gets worse with time, and eventually it leads to paralysis. There's also difficulty breathing because of the diaphragm and intercostal muscles. So it's a lethal disease in many cases. So the death can result from pulmonary respiratory failure, from pneumonia, infections, things like that. So it's a devastating disease, and it's also a genetic disease, and it affects approximately 1 in 10,000 newborns.
I just want to let our listeners know that you are listening to My Hometown on the voice of Nassau Community College, 90.3 WHPC. I'm Nassau Community College student Zach Turkel, along with Bill Haran, and our guest today is Dr. Adrian Krainer, who is a professor at Cold Spring Harbor Laboratory. Bill, you have a question, right?
Yes, I do. But first, I want to thank you, Dr. Krainer. You know, most of us, when we see someone in the military or a policeman, we go up and nowadays we thank them for their service.
Right.
We may not even know that it's your discovery that cured somebody or had an important role in keeping someone from getting one of these terrible diseases. So just to start off, I want to say thank you. When you came up with this breakthrough, was it one of those eureka moments? Did something just happen, or was it by mistake, or did you wake up one night in the middle of the night and say, that's it, move column A to column B? How does that, in a breakthrough like this, how does it come about?
Yeah. First of all, thank you very much. And so that's a good question. There were, I would say, multiple eureka moments, but it is a long process. So drug discovery is something that takes years, and we began to work on SMA around 2000. And before that, we had already spent many years investigating the basic mechanisms of RNA splicing. And once a connection was made between this disease and an underlying defect in splicing of RNA from a particular gene, we began to work on that. And So there's a series of experiments, at first dealing with basic aspects, and eventually ideas began to crystallize for how you might develop a therapeutic to correct the splicing defect. And we began the particular pipeline that led to this drug. We began working on it and working on that in 2004. And this was work that was done in collaboration with Ionis Pharmaceuticals, a company in San Diego. And my main collaborator there, Frank Bennett, was a co-recipient of the Breakthrough Prize with me. And we worked on it for something like 7 years here at Cold Spring Harbor, doing all the steps. I would say that the work proceeded very smoothly, even though it takes such a long time. So every step along the way seemed to work rather well. Typically in science, you often have a lot of setbacks, and sometimes you reach just an impasse and you can't make any progress. And that wasn't the case here. Everything sort of moved along, and it was fairly clear what the next steps should be. And although there were a lot of difficult experiments, thanks to many talented people, everything worked nicely. And then by 2011, it was time to begin to test this experimental drug in patients, and so that's something that Ionis Pharmaceuticals undertook, and it involved clinical teams in several countries, and so that's when clinical trials began. I would say getting to that point was a very big milestone. And then the fact is that these trials worked rather well, and again, that's a process that took 5 years, and that's considered fairly fast in drug development terms. And the results were very clear, and the drug worked like a charm, and so the results in many ways were better than expected. So it was a very fortunate situation, and this led to rapid approval by the FDA and other organizations in Europe, etc.
Must be the ultimate high, Zach.
Yeah,
yeah, definitely. Yeah, it's just we're scientists. We're happy to be in the lab and trying to find out how our cells or our genes work, and in a way, it's solving puzzles. But you always hope that this work can have a dramatic impact in people's health or other aspects of society. The thing you never know is that if that will happen, when will it happen? It might not be in your lifetime. You could just be contributing and enabling somebody else to make the next discovery, and so that's always the case. Having this produce such dramatic results and saving lives and improving people's lives, so relatively rapidly is indeed the ultimate high. And by now there's about 6,000 people around the world who are receiving this treatment.
So, Dr. Krainer, the FDA has approved the use of this drug to treat the disease, which I am sure is a big deal. And what's involved in getting that level of approval? Does it take a long time? How does that process work?
Yeah, so I wasn't directly involved in that, but of course I followed it with interest. So here at Cold Spring Harbor, we don't work directly with patients. This is a basic research institution, but what's involved there, it's a very careful process. After having clear data in the laboratory with cultured cells, with experimental animals, and a lot of safety data. Then there's a process of engaging in a dialogue with the FDA to begin to propose how clinical trials will be conducted and what are all the safety measures that are in place, and what exactly will be measured in these trials, how many patients should be involved, and for how long. So there's a lot of details. So typically these applications are very complex, and so there's a dialogue, a lot of back and forth. And then clinical trials begin, and so there's a lot of reporting and discussions to sort of adapt to the observations. And clinical trials are done in phases 1 through 3, and the initial results inform the larger trials that follow.
Dr. Krainer, it's certainly very interesting, and we want to find out more, but this is a great time to take a break. I'm Bill Haran, along with Zach Turkel. Our special guest today is Dr. Adrian Krainer, speaking about RNA splicing. You're listening to My Hometown on the voice of Nassau Community College, 90.3 WHPC.
Hi, everyone. I'm Kristen Bell, and I invited you here to my hotel room to talk about your cheeks. Your big, beautiful cheeks. Get your mind out of the gutter. I'm talking about swabbing them. Did you guys know that each year 20,000 Americans with blood cancer are searching for their life-saving marrow match? Can you even believe that? I mean, people need marrow, you guys, and we have it. So here's how we can help. It's really, really simple. All you have to do is swab your cheek. Then go to Gift of Life Marrow Registry. It's so easy, anyone can do it. And in case you're wondering, if you're a match, donating, it's a lot easier than you think. So wherever you are, go ahead and swab that cheek. Go to giftoflife.org and get a free kit sent to your home, or find a donor drive in your area so that you can swab a cheek and save a life. It's only a minute of your time, but it can give someone else the rest of their life.
My Hometown is made possible by Anton Media Group. For more of your local community news, visit AntonMediaGroup.com. If you have a story to share or want to subscribe, call 516-747-8282.
WHPC. And now we return to My Hometown.
We're back on My Hometown on the voice of Nassau Community College, 90.3 WHPC. I'm Bill Haran, along with Nassau Community College student Zach Turkel, and our special guest today is Dr. Adrian Krainer, talking about RNA splicing. Dr. Krainer, can you tell us about when you get, basically what you got is like the Oscars as we're familiar with on TV, but this is an Oscar for science. What is that like when you get that award? Do the people fight over it? Do they give you a certain amount? Is it proportional X dollars every month? Or what happens behind the scenes?
Well, I didn't know what to expect, but it was a pretty exciting and interesting experience. So I got a call one day from a scientist that I highly respect who was a previous recipient of this prize. And so she informed me that I and my collaborator were selected for one of the awards this year, specifically one that of the Life Sciences Awards focused on neurodegeneration. So that was a great surprise. It was very exciting, and it was very exciting to get the news from her, Dr. Huda Zogby, in this particular case from Baylor College of Medicine, rather. And but that was several months ago, and we have to keep this secret. And then there's very detailed preparations for the awards, for the events surrounding it. And then my wife and I flew to San Francisco, where this all happened, and we had quite a bit of information along the way, in the intervening time, to tell us what to expect and all the aspects of the ceremony itself. We also met with some of the sponsors of the prize, who are like Zuckerberg and Chen and Yuri Milner and the founder of 23andMe and the founder of Google, Sergey Brin, and Dr. Wojcicki and several others. So we had an opportunity to meet with these people, and there were some interesting dinner parties. It was a lot of activity for 3 or 4 days, and we met other laureates, including many of the laureates from previous years. And so that includes a lot of scientists that I knew before or I knew of. But it wasn't only life sciences, so we also met with physicists and mathematicians, so it was pretty exciting.
Zach?
And Dr. Krainer, if anyone is listening and they're very interested in this, and if they want to learn more about the information that you've researched, how can they get in touch with you?
Right. So Cold Spring Harbor Laboratory, so there's a website, cshl.edu. So there's a lot of information about the research that goes on in this institution. There are also educational activities at all levels. This is a center for molecular biology research and education. And you can see there the different faculty members of researchers, and I'm one of those. So there's information on the website and also contact information.
And we'll give it out towards the end of the show again, just for our listeners that just tuned in as well.
Dr. Krainer, if someone has this disease or a member of their family, is there some way they can find out more information and just where they could get their hands on this drug, or can they? Is this too early in the process, or is there some way some of this scientific discovery can help them along their trails?
Yeah, so the drug was approved at the end of 2016, so it is already available and used around the country and in many countries around the world. I would say, obviously it's a prescription drug, and so people would find out about its use through their physician, their pediatrician, or neurologist, typically. There's an organization called Cure SMA, one word, curesma.org, that has a lot of resources and information for families that first encounter this disease. So that can be overwhelming, but besides talking to their physicians, Cure SMA provides a lot of resources and of course, information about this drug and other experimental treatments and resources.
And my question is, Dr. Krainer, that, let's go back to this breakthrough that you made with the disease spinal muscular atrophy and the new drug that came from your research. What made you want to study that particular disease in the first place?
So my entire career, scientific career, has focused on this process of RNA splicing. So, like I mentioned before, we have studied what it is, how it works, the details of how it goes on in our cells, and why our genes involve this process. We were very aware of the relevance of RNA splicing for both healthy and diseased states. So we investigate, for example, the role of splicing malfunction in the context of cancer and various genetic diseases. When I came across SMA around 1999, 2000, it was apparent by then from work in other laboratories that the disease was caused by defects in a gene, and that there's a gene present in patients where splicing isn't quite as efficient as it should be, and so unlike other diseases that we were studying at the time, it seemed that this was a case where if we could develop a therapeutic, it would potentially help all the patients with the disease. In other diseases, you have different mutations, and some of them affect splicing, but the majority do not. And so it would be a case of having to come up with more personalized therapeutics, which it is, I think, the way to go in the future. But in this particular case, it was very attractive because all the patients have the same gene, the same defect that you can target in the same way.
And I just want to let the listeners know that you are listening to My Hometown on the voice of Nassau Community College, 90.3 WHPC. I'm Nassau Community College student Zach Turkel, along with Bill Haran. And our guest today is Dr. Adrian Krainer, who is a professor at Cold Spring Harbor Laboratory right here on Long Island.
Dr. Krainer, since we're broadcasting from a school, someone hearing this may say, gee, that sounds so interesting, and I would like to do something, or at least attempt to, that could help humanity. If someone is interested in getting more into science, maybe at the high school, college level, what are some of the courses? What direction or advice could you give them?
Yeah, well, if they want to do something similar to what I did, or study RNA splicing in particular, I, for example, studied biochemistry in college and in graduate school. The relevant coursework is certainly biology, chemistry, biochemistry, Yeah, so that's kind of a way to get a handle on it. Early in my career, I was fascinated by genetics, the classical genetics of Gregor Mendel, and so that's what kind of got me interested in biology and genetic diseases, and later molecular biology and molecular genetics.
Have you kept in touch with families afflicted with this disease, and what feedback have you heard from them?
Yeah, very much so. During the entire process that we worked on developing this drug and understanding the defects in spinal muscular atrophy, I've been attending annual meetings that bring together patients and families, clinicians, and researchers. And so I've made many good friends in the SMA community. Some patients are right here on Long Island, and we're frequently in touch. And so the feedback has been really amazing. I would say the very evening that the drug was approved, it was 2 days before Christmas in 2016. So I got emails, in particular, an email card signed by dozens of patients and their families who were very excited that now the drug had been approved. I've met many patients who participated in the clinical trials that made it possible to get approval for this drug, and others who have begun the treatment subsequently. And so I've met very young patients. Some I've known for several years, so some of those are already adults or children, and it's amazing to see the types of improvements. But I've also corresponded with adult patients, and again, they tell me what difference they're experiencing not so long after initiating treatment, so it's extremely encouraging. For instance, I hear of people who, there was a rider, but he's 38, and he said at this point in his life he could only move his thumbs, no other muscle in his body. But, he's very creative. He's an artist and a writer. And so now he says that he began to be able to move several fingers that he hadn't been able to do that for 15 years. And that many patients with that severe form of the disease need ventilatory support to be able to breathe. And so he was only able to not use that machine for four hours a day, and after a few weeks with treatment, now he's increased that time without the respirator to 6 hours a day. And so one aspect that's very encouraging is people are seeing improvements to different extents depending on how severe the condition is, how many years they have had it. And these improvements appear to continue over time. So not only do you stop the progressive degeneration of SMA, but we are seeing continued improvements over time. So that's really very, very encouraging.
Dr. Krainer, if you wanted to leave us with one piece of wisdom from yourself, an award-winning scientist who lives and works right here on Long Island, what would that be?
I would say for a scientist, at least, that you have to love what you do. You get it's a way to satisfy your curiosity, but it's also an opportunity to contribute to knowledge, but sometimes in a way that can lead to a very practical application from which many people can benefit, and so it makes it all worthwhile.
Dr. Krainer, we want to thank you so much, and let our audience know again, our guest was Dr. Adrian Krainer, the professor from Cold Spring Harbor Laboratory, for his discussion on spinal muscular atrophy, a neuromuscular disease that's a leading genetic cause of death in infants. He's done a great job, as you've heard. He's made life a lot better for people. Thank you so much for being a guest and for the great work. Please keep it up. I'd like our audience to know that I'm Bill Haran, along with Nassau Community College student Zach Turkel. Thanks for listening to My Hometown.